Hello, and students. Welcome to TSA's Tutorial Center. In this tutorial, we talk about molecular biology. Okay, so what is molecular biology? That's what we're going to explain today. We'll do the introduction to molecular biology, and we'll also talk about the terms which are used. We'll also talk about mitosis. Then in the next video we'll talk about meiosis, and you see how simple we're going to break them down in a way that you never forget. Okay, so the question is, what is molecular biology? You can define molecular biology as the branch of biology that studies the composition, structure, and interaction of cellular molecules. Very simple. Now, the cellular molecules that you'll be talking about for most of the time will be just the nucleic acid and the proteins, because they are the one which carries out most of the biological processes in the cells. Now, from there, you're supposed to know, to just have an idea of this topic that you've been talking about when you're in grade 12. Do you still remember genetics, or you have forgotten? Genetics. I'll give you 3 definitions. You can't forget 3 definitions. The first one, you can say, this is the study of how traits are transferred from one generation to another. Repeat, this is how genetic information is transferred from one generation to what? To the other. The second one, we may say, this is the study of how traits are transferred from the parents to the offspring. This is how traits are transferred from the what? From the parents to the what? To the offspring. The last one, we can say this is the study of heredity and variation of inherited traits in living organisms. Now, you can't just say this is the study of heredity and variation, and yourself you don't know what it means when we say heredity. What is heredity? This is the tendency of an organism to resemble its parent. The same way you look alike with your parents. Kaya, if you do look alike, but you're supposed to, just ask them, or you just get a mirror, you observe if you look like one. Then variation, these are the differences in the characteristics of organisms of the same species. That is, you're supposed to compare man to man. You can't compare yourself to a dog. You're supposed to compare you with a human being. Now from there, it is worth mentioning that we're going to talk about the cell cycle. That is where we're going to have the mitosis and meiosis. So the cell cycle. What is the cell cycle? Okay, so this is the sequence of growth in DNA replication in cell division. Now it's weird. In 1 hour, in your body, about 1 billion cells are destroyed and 1 billion cells are created. Look how powerful God is. The cell cycle is actually divided into 2 basic phases. That is the interphase, which is divided in G1, S, and G2 in 3 phases. When we say phase, we're talking about stages. Then there is also the M phase. We're saying the cell cycle is divided into 2 basic phases. That is the interphase, which is divided in G1, S, and G2. There is also the M phase, which is divided in karyokinesis and cytokinesis. Kari means the nucleus. Kinesis means to break down. So the breakdown of the nucleus is called karyokinesis, and the breakdown of the cytoplasm is known as cytokinesis. You can even name your firstborn. Your firstborn, cytokinesis. The second one, karyokinesis. I love the names. Let's go to the second thing we should know. It's very much important to talk about the terms which are used whenever you are studying something. Let's talk about the terms which are used. What are chromosomes? This is a thread-like structure found in the nucleus which store genetic information in form of DNA. Very simple. Now, the same chromosomes, there might be sex chromosomes and autosomes. Now, when we say sex chromosome, we are not saying they look sexy. No. Sex chromosomes, they're just associated with sex. That is the sperm and the ovaries, the ovum. Then the autosomes, this is any chromosome apart from the sex chromosome. Any. If it's not just the sex chromosome, it is known as autosome. Then there is also these kind of chromosomes which are known as homologous chromosomes. We'll talk about homologous chromosomes very much when we start talking about meiosis. Homo, same. So they're going to have something which is in common. These are chromosomes that have a gene in the same position, same shape, and same size. Then you're going to also talk about replicated chromosomes. Now, chromosomes, replicated chromosomes, they are chromosomes formed after replication, and they are made up of two chromatids. Suppose you have this is your chromosome. When it replicates, they're going to be two, and they're going to be called chromatids. Now, they're going to have the center here, which is known as the centromere. When they combine at the centromere, they are now known as sister chromatids. Okay, sister chromatids. So let's talk about the sister chromatids. The sister chromatids are two identical chromatids formed when chromosomes do what? Replicates. Each has a double strand of the DNA, and they are actually joined on the centromere. Then there is the bivalent. Bi means two. So when you have two homologous chromosomes, then they pair up. That is known as bivalent, which will come and talk about a lot when you start talking about meiosis. Have you ever heard of haploid, diploid? There is also polyploid. What is a ploid? This is the number used to represent a set of chromosomes. When you say haploid, hap means one set of chromosomes. When you say di means two, so two sets of chromosomes. Then ploid means more than two sets of chromosomes. Then according to what we said, we'll talk about sex. The sex or the gamete cells involved in individuals. Okay, so for the mother and the father, the chromosomes, they have been given the name. I'm talking about the mother, they are known as maternal, and the father is known as paternal. Oh, you are here, paternal. Okay, so as we go on, we'll talk about spermatogenesis, which is very interesting. Um, just in case of cases, they might ask you to determine or to mention how many chromosomes are there in the mains. So take note of these notes. In human, we know that there are 46. In maize, it is 40. Rice, 24. Tomato, 24. Onion, 16. And Drosophila, they're just 8. And from there, we're talking about the cell cycle. The cell cycle has checkpoints, which is known as the cell cycle control. There are basically 3 types of checkpoints. The first one is G1, the second one is G2, and the third one is M checkpoint. Let's talk about the G1. G1 checkpoint. So the first one, this is more like the first checkpoint which is going to be there. It's going to check if the cell has fully grown and is ready for division. More like your parents looking at you like, this one ready for marriage. Nah, no, still young. That is the G checkpoint, G1 checkpoint. It also checks if the environment is suitable for the next stage. Then, the G2 checkpoint. We said the interface is divided in 3. There is G1, S, then G2. In the S phase, that's where the chromosomes in the cell are going to replicate. Now this guy, the G2 checkpoint, will come in and it's going to check if the replication of those chromosomes was correctly replicated. If not, there's going to be corrections. Then the M phase. Okay, so the M phase. Okay, so to understand the M checkpoint, you're supposed to understand the M phase. Suppose this is your cell, plant cell. And this, their sister chromatids. There is the centromere there, centromere there, centromere there. On the centromere, there is a protein which is known as kinetoco. Don't incite your neighbor. Kinetoco, not cadinetaco. Kinetoco. The purpose or the function of the kinetoco is they combine or they bind. They are the one which causes the mitotic spindle or fibers or the microtubules to bind to the chromosome. So the M phase is going to check if all the chromosomes and the kinetoco is attached to the microtubule. These kind of ropes here are the one which are called the microtubule. Okay. Now, let's talk about now the proteins which are responsible now for the progression of the cell through all the stages. They are known as cycling proteins. In definition, we might say these are the proteins that are responsible for the progression of the cell through the stages. Now, there are just 2. There is normal P53 and abnormal P53. The normal one, what it does is, when it says that this cell, it is damaged, it will call the police. But the police in this case, they are known as apoptosis. Apoptosis Apoptosis is to terminate. Is the process. Well, have you ever watched this movie known as Terminator? Yeah, think of the P-53 as the Terminator. It will go upon apoptosis. Then the cell which is damaged will be destroyed. Okay, so this is what we are saying. Detects DNA damage and initiates repair or triggers apoptosis. Programmed cell death. The cell will be killed. If the damage is irreparable, then preventing damaged cells from dividing. So it will stop dividing and it will be killed. Then from there, we said there are two. So there's going to be another one which is known as abnormal B53. Now the abnormal one, look at it. The abnormal one, it doesn't go upon apoptosis, no. It doesn't trigger apoptosis. It will be just looking at the cell which is dividing, minding its own business. I love this one. But if the cell is not terminated, then that's where we're going to find that the child that has come to life might have cancer, or sickle cell anemia, any kind of genetic disorder. So it allows the cell to continue dividing, minding its own business. So sometimes minding your own business is bad. Just go put your nose in those kind of business. They tell you, don't mind our business, just go put the nose in. Boom. Next, familiar yourself with these terms which I used. The chromatin, it is also known as the chromatin. This one has no name, other name, it's just known as the chromatin. Molecule made up with the DNA and the histones. You should go through this chromosome. We have talked about that. And the centromere, the cohesin protein. I showed you where it is positioned. And the kinetochore, that is the spelling. And the pronunciation is kinetochore. Kinetochore. I said don't insult your neighbor on this one. Anyway, the microtubules, those tumor-ropes which were joining or attaching to the kinetochore, very much important. Now let's talk about the mitosis. What is mitosis? It is worth mentioning that mitosis is also known as equational division, because the number of chromosomes of the parent is going to be equal to the number of chromosomes of the offspring. Now, when they tell you to say, what is mitosis? You might just say, is where a parent cell divides to form two identical daughter cells. That is mitosis. Now, mitosis, it will be divided into two basic stages. The one we talked about, the interphase and the M phase. Now, let's talk about the interphase. We said the interphase is further divided into how many? 3. That is the G1, G means gap, the gap 1, gap 2. But between those gaps, there is the S phase. Let's start with the first one, the gap 1 phase. Okay, so in this stage, the cell is metabolically active. It continues to grow as you are growing as well, but it will reach a point where it's supposed to give birth as well. I'm sure you have reached. You are just waiting for the right time, but continue waiting for the right time. It's very much important. Focus on school, my dear. So the cell continues to grow and produce proteins and organelles, but DNA replication is not going to take place. Then from there, we said between the gap 1 and gap 2, there's going to be the S phase. There we go. What happens in the S phase? That's where the DNA replicates. Now take note, the number of chromosomes remains the same, but the DNA doubles. So if a cell, for example, if a cell has 2C DNA at G1, after the S phase, it will be 4C. Now, maybe if you don't understand this, let me try to explain. Suppose you're trying to photocopy a book which is made up of 2 chapters. These are the number of chromosomes. These are chromosomes. When you photocopy the book, you're going to have the same number of chapters, but the DNA, they're going to be how many? 2. Have you gotten the concept? This is your book. The number of chromosomes will remain the same number of chromosomes, 1, 2, 1, 2. You're just going to have 2 chapters. It's not going to be 4 chapters now. It will be 2 chapters. The same information which was on chapter 1 will be the same information which is going to be here. That's what I'm trying to say. Now let's talk about the G2. Now here it's known as the G2. The cell now is ready now to replicate, to divide, ready for mitosis. Prepares. It produces proteins such as spindle fibers, centrioles, and the microtubules. And the cell continues to grow in size. Then, when all this happens, then we go to the M phase. And I told you, this one, I love it. The M phase, we said, is divided into 2. There is a karyokinesis and a cytokinesis. Karyo, what does it mean? It means nucleus, and cyto means cytoplasm. Kinesis means division. Okay. So we'll start with karyokinesis. Now in karyokinesis, there are actually 4 stages. I used to make my small mistakes when I'm talking about the same 4 stages. Maybe I'll start with the first one. The last one, I make it the first one. So I came up with a word which you may be using. I call it prominent. Prominent. This is A. Prominent. This is A. Prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. Okay, so we'll start with the prophase. Prophase. This is where because things will start appearing and things will start disappearing. The cell chromosomes condense and becomes visible. They condense, they coil up and becomes what? Visible. The first one. The second one, the nucleus breaks down. In some books, they say the nucleus disappears. The nuclear membrane starts to disappear as well, to expose the chromosomes into the cytoplasm. So to expose the chromosomes to the cytoplasm. Then from there, the mitotic spindle, the microtubules will do what? Will start appearing. They will form and extend between 2 poles. Then from there, we said prominent. So we're supposed to go to the metaphase. Now, before the metaphase, there is this quick phase between the prophase and the metaphase, which is known as pro-metaphase. So it is having two stages, prophase and metaphase. Now this stage, this is where now the kinetochore will start being attached to the microtubules. Boom, 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 boom. They start to attach to the microtubules, and it will be considered as done or completed when all the kinetochore are attached to the microtubules. Then we can go to the metaphase. Now the metaphase, it's very simple. You have chromosomes. Meta means at the center. They will start lining up at the center, which is known as the meta plate, at the center of the cell. And there is the centrioles having the microtubules. This is the metaphase. So the sister chromatids will come now at the center of the cell, and their kinetochore will be attached to the what? The microtubules. The anaphase. The anaphase, it is the shortest. What happens is this. You have this as your chromosomes. I'll just draw two. Then this is the cell. There is the microtubule and the microtubule. It is worth mentioning that where they were joining the sister chromatids, it can break down. It can break down where they were joining. It's known as what? The centromere. So the centromere can break down. At the anaphase, the microtubule pulls this sister chromatid. It will pull this one in this one, then they will break like this. Then move to at the end or the poles of the what? Of the cell. So even to this one, exactly the same thing that's going to happen. That is the anaphase. Okay, so we are saying the centromere breaks down, hence the sister chromatids are separated and pulled to the opposite poles of the cell due to the contraction of the water, contraction of the what? Microtubules. Very simple. Then the telophase. The telophase is also known as the opposite of the prophase. Let's talk about it. The first thing, the chromosomes, we said they coil in the prophase. In this stage, they are going to do what? They're going to uncoil and becomes invincible. It becomes invincible. So here they were supposed to be invincible. This is wrong. Then what else? We said the nucleus is going to do what? The nucleolus is going to do what? Disappear. In this stage is going to appear in the mitotic spindle, which appeared in the prophase. It is going to disappear in the telophase. And finally, the chromosomes will appear at the end poles where the cell nucleus, the cell nuclei forms surrounding the chromosomes. So something like this. But we said the chromosomes have started moving to the end poles. When they start reaching this far, the nucleus is going to form. The nucleus is going to form. They will surround. They will surround the chromosomes. This is the cell. Then finally, this thing is going to break. The cytoplasm is going to break down. And we said the process is known as what? Cytokinesis. cytokinesis. Let's talk about cytokinesis now. Okay, so we say this is the separation of the cytoplasmic contents to form two daughter cells. And let's talk about it. Cytokinesis, however, differs in animal cells and plant cells. In animal cells, it's done by the action of actin filament. Now, this actin filament just inside the plasma membrane, that's where it's going to form. Inside the metaplate, the actin filament it's going to pull the center of the cell inward. Going to pull the center of the cell inward, something like this. Inward. Then when it pulls the cell inward like this, it's going to form the cleavage furrow. This one, it will deepen, it will continue to deep, becomes tight, and finally the cell is going to be broken into two daughter cells. What about in plant cell? In plant cell, it is actually very simple. In plant cell, do you remember the vesicles? Yes, the vesicles from the Golgi body containing cell wall material. They're going to do what? They're going to gather at the former plate to form the cell plate shaped structure. To explain this better, it's more like this. There will be no formation of the actin filament because of the cell wall. It's very difficult to break down this one. So, from the Golgi body, the vesicles will start to be produced. Then they will come and line at the center of the cell. They will continue to be there. Then when they emerge with the cell wall, then the cell is going to break down. So next time we're going to talk about something interesting in biology. It will be meiosis. We'll define what meiosis is, myogenesis. We'll also talk about spermatogenesis and many more that you're supposed to know. So keep in touch. Thank you for watching the video.